Okay, so let's talk microbial metabolism. I don't know why it didn't load as a PowerPoint, and I don't have another copy, so it's a PDF, so just sorry. Okay, so what is metabolism? Metabolism is all chemical reactions that happen in a cell. This includes building and breaking. Okay, so the term anabolism means to synthesize or build something. I can take individual amino acids, individual Legos, and build a protein and build a Lego castle. Okay, that's anabolism, going from pieces to something big. Catabolism, catabolism, okay, is breaking down. I think of my cats. They don't really build anything, they tend to break things. So, this is taking the steak that you've eaten and breaking it down into component parts. So, anabolism is a side of metabolism where it assembles small molecules into larger ones that are for the cell to function. Energy is required for this process to happen. You're not going to build a house without using some type of energy. Catabolism is a side of metabolism that breaks down macro, big molecules into smaller molecules, basically yielding energy. Energy that is produced and used is in the form of adenosine triphosphate. Again, this is our fuel. Adenosine triphosphate is where we store our energy, and when we break it, we get the energy back out. So, I kind of discussed enzymes a little in the last chapter, but not a lot. So, enzymes are proteins and they are known as catalysts. Basically, they increase how fast chemical reactions happen and they decrease the amount of energy required for that chemical reaction to happen. So we say they help to catalyze reactions involving reactants, which you can also call a substrate. Substrates link to your enzyme's active site. Okay, so the enzyme has little slots for those substrates to bind to. Um, and then the enzyme puts them together or tears them apart. So imagine me taking a nut and a bolt and putting both of them into a mayonnaise jar and shaking them and hoping that they spool together. How long do you think it would take shaking that mayonnaise jar to spool a nut and a bolt together? Probably a long time, right? It's going to take years. Most chemical reactions that happen in your body, if we counted on random chance, like the shaking mayonnaise jar, to get things put together or even taken apart, if I put them together, put them in a mayonnaise jar and shake it, how long until it unscrews? Yeah, it's going to be lengthy. Most chemical reactions that happen in your body, most of the metabolism that happens in your body would be like that mayonnaise jar. If we counted on random chance that this would happen, that would take not only a lot of time, but would take a lot of energy of shaking that mayonnaise jar and hoping that by some miracle, the right thing happens at the right time to get it to either unscrew or screw back together, right? Enzymes are like taking your hands, getting the nut, getting the bolt, putting them together. How much energy did I use to make that happen? Not a lot. Compared to the shaking of the mayonnaise jar for 10 years and it randomly happened. Because of that, enzymes are essential. Most of the chemical reactions in your body would take so long you would never live. Having enzymes gets them to happen fast and for a lot less energy than shaking the mayonnaise jar for 10 years. Okay? So, enzymes themselves are proteins, they're made of protein. Simple enzymes are normally protein all by itself. Conjugated enzyme. To conjugate means to bring together. Are proteins usually combined with a non protein cofactor or coenzyme, usually derived from vitamins. Remember, I talked about the ignition key. Enzymes are expensive. So when I don't need it, I just want to turn it off, put it to the side, and when I need it, I put the key back in rather than making new every time. That's what this is. A conjugated enzyme has an ignition key, okay? Enzymes have particular characteristics that you need to be familiar with. So this is table 7.1. Most are composed of protein, and they may require cofactors, an ignition key. They act as organic catalysts. Catalyst is something that causes a reaction to speed up the rate of cellular reactions. Me using my fingers versus shaking the mayonnaise jar. It speeds it up, it makes it go faster. They have unique characteristics such as shape, specificity, and function. You will not see an amylase enzyme, which is a starch enzyme that breaks down starch, working on lipids. You won't. They have one specific job, and that's it. So they have specificity. The shape that they are in is also very important, because if I want to grab the nut and the bolt, I'm not going to be able to do that with my back. I have to have the right shape so that I grab the things that I need to grab. And function. Am I putting stuff together? Am I taking it apart? What's my job? I have a specific job if I'm an enzyme. They enable metabolic reactions to proceed at a speed compatible with life. Again, if we waited the 10 years for the nut and bolt to come together, that's a long time. If I need to convert oxygen and glucose into energy, I don't have 10 years to do that. I can't not eat and not make energy for 10 years. It would kill me. So having these sped up reactions is compatible with life. It helps my body to survive. It helps your body to survive. It helps everything to survive. They have an active site for the target molecule called substrate. Like I said over there is a binding site, an active site for these things to actually, the nut and the bolt to actually fit into. They're much larger than the substrate. If I'm going to manipulate the shape of two things, I have to be bigger than those two things. A nut and a bolt are not going to be put together by an oxygen atom. They're a lot bigger than an oxygen atom. You have to have the right, I guess, size to be able to manipulate the two things that you are putting together. Okay? So you can't, how to put it, you can't be small. You have to be bigger than what you're working with. Let's see. They associate closely with substrates, but they don't become integrated into the reaction products. When I put the nut and the bolt together, my hands didn't fuse with the nut and the bolt. They didn't fall off because I touched the nut and the bolt. They didn't die because they put the nut and the bolt together. Enzymes are the same. They come, they put the thing together, they go and look for two things to put together again, the same things, but they're looking. So if I'm an enzyme, I put the nut and the bolt together, I float off and look for another nut and bolt to put together, because that's my specific job, and I can do it over and over and over again. I'm not used up. I don't become part of anything. Literally, I just do my job over and over and over. Okay? So they don't become integrated into the reaction products. Also, they're not used up or permanently changed. Again, my hands didn't like fall off. I didn't like lose the ability to put nut and bolts together. Same thing with the enzyme. They work over and over and over again. Remember, I said enzymes are expensive. I don't want to have it be a one-trick pony where it comes in, does it once, and then it dies. I just spent $100,000 creating this enzyme. It better work more than once. Okay? They can be recycled, thus function in extremely low concentrations. If I don't need it anymore, I can break it down and use it to make some, like the component parts, to make something new. And because they're not changed and they work over and over again, I don't necessarily need 100 million. I might only need 100. They're greatly affected by temperature and pH. So remember, I said that proteins, especially at different temperatures, will unravel, like the sweater, right? I said the knit sweater will unravel and it won't come back together. Well, an enzyme is a protein. It has movable parts. It's a complex structure, which most proteins are. So when it comes down to it, if you change the temperature enough in a body or in a cell, the proteins will, just like the sweater, unravel. pH plays a role as well because we have something called hydrogen bonding. Remember, I talked about the stitch with the disulfide bond? It's a little bit similar to that in the fact that hydrogens bond and create the three-dimensional shape that you need for that enzyme to be the right shape to do the job you want it to do. If you start changing the pH, either you're going to start inserting hydrogens or you're going to start taking them out, and that's going to cause it to unravel as well because those stitches that were holding it in the right shape are no longer there. Okay. They can be regulated by feedback and genetic mechanisms. So if you have enough of a product, sometimes that product comes back to the enzyme that created it and docks, basically making it not work anymore. It's a way to stop it from working because it's a feedback. I had something, I made it, it came back and stopped the enzyme from working. Okay. Now, enzymes are classified and named according to what types of reactions they catalyze. Groups of enzymes are usually going to have a prefix related to some type of reaction catalyzed and combined with the ending ACE, ASE. If you see the ending ACE in biology ever, it automatically means it's an enzyme. And the name often tells you what kind of enzyme it is. Okay? So, oxoreductase. Um, they transfer electrons from one substrate to another. So, redox reactions or oxidation reduction reactions are literally somebody's losing electrons, somebody's picking them up. So, oxoreductase, oxidation reduction, ACE, it's an oxidation reduction enzyme. Electrons are being moved from one and another thing is picking them up. Transferase. What do you think it's doing? It's transferring something. So you've got a transfer of a functional group, a piece, okay, a group of molecules from one substrate to another. So I'm taking this piece off of this one and putting it on this one, okay? Hydrolase. Hydro, water, right? So it has to be an enzyme dealing with water. So breaks bonds on molecules by adding water, okay? Lyase, add or remove groups from double bonded substrates. Isomerase rearranges groups within a substrate, so it actually changes the shape of something that already exists. Ligase, to ligate means to bring together, like to glue together. So it catalyzes the formation of bonds with the input of ATP and the removal of water. So it's not necessarily free, remember. I just bring the amount of energy needed down. Okay. When I put the nut and bolt together, technically I am using energy to get my muscles to work and to do all of that. It's just less energy than shaking a mayonnaise jar for 10 years. So in this case, I actually even mention it takes ATP to get this to happen. All right, so factors that affect enzyme function. The cell's environment can affect the enzyme's activity. High temperatures, like I said, abnormal pHs, even changes in water pressure, osmotic pressure, can cause the enzymes to become unstable. Sweater, unraveling. So we call that denaturation. Okay, so denaturation is when the bonds within the enzyme break and the enzyme is permanently useless. Again, unravel a sweater, I'm not going to be able to put that back together. Okay, inhibitors can affect enzyme activity as well. And we kind of have two types of inhibition when it comes to enzymes. We've got competitive inhibitors. Competitive inhibitors are going to occupy the active site that our substrate normally goes into. Okay, so our substrate goes in and let's say in this case, our substrate goes in, we break it apart into two different things. We take the nut and bolt apart. A competitive inhibitor actually bonds to that active site, preventing my substrate from going in. So it's competing with the same placement. Okay, that's why we say it's competitive. It prevents the substrate from binding. Our reaction doesn't occur. Antihistamines are competitive inhibitors. They bind to the histamine receptor and don't allow histamine to get to it. And the fact that you've got antihistamine means that that reaction you're looking for doesn't work, okay? Or your body's looking for, not necessarily you, because if you're taking an antihistamine, you're trying to stop something, right? Okay, non-competitive inhibitors. They inhibit the enzyme activity by binding at a regulatory site. Notice that didn't say active site, okay? So not where your substrate binds, it's another place on that enzyme, which changes the active site and prevents the substrate from binding. Many metabolic pathways are regulated this way when enough product is produced. So here is my normal substrate. I make this product. Now remember we talked about feedback, right? Once I have enough of this product, it'll come back and bind to the enzyme. See how the active site isn't the same shape anymore? That means this guy can't bind anymore. I'm not making any more of this product. So once I have enough, it's a good off switch. I've got enough of that product. I don't need any more. Okay, let's make it go back and bind to this thing so that it'll stop making it. Okay, so it's a feedback. It comes back around. Okay. So redox reactions. What helps generate ATP? All right, so electron transfer through oxidation reduction reactions helps to generate ATP. Electrons get transferred from one thing to another, okay? Excuse me. So one of the ways that I remember this is oil rig, okay? Oxidation is loss. Oxidation is loss of electrons. Okay? Rig, reduction is gain. So reduction is gain of electrons. Okay? Now, normally in real life, you don't have one without the other. If somebody's losing electrons, somebody's gaining electrons. So we don't necessarily refer to it in real life as an oxidation reaction, the end, or a reduction reaction, the end. If there's an oxidation happening, there's a reduction happening. If there's a reduction happening, there's a oxidation happening. So we tend to use the term redox where they're combined because, like I said, one doesn't really happen without the other. So redox always occurs together. One substrate loses electrons, the other gains those same electrons. And we call them a redox pair because I gave them to you. You're the one who accepted them. Electrons don't just fly around the cell. They're shuttled, actually, by electron carriers. So think of them kind of like electron taxicabs that carry them to a specific place, known as NAD plus and FADH. These shuttles load and unload the electrons from one substance to another. So I pick up from substance A, I drive it to substance B, and I drop off. Okay, like I said, taxicab. It's a taxicab that picks up and drops off. The movement of the electrons will eventually generate more ATP. It'll generate more of our fuel source. Okay, let me see.